Hi, this is Miss Linton, and this is my wonderful period 135 review. Say hi. Hi. All right, so what we're trying to do is do a kind of a rewind just to keep things fresh in our head. So we're ready for any kind of ghost questions on exams, yes? And so I wanted to remind us of Unit 1. The first chapter was a view of life. That was kind of the basics. Then we went into basic chemistry, and then the chemistry of organic molecules and cell structure and function. So I just wanted to do a brief review on Chapter 1 and on Chapter 2 today. And then I embedded some questions in there in Hello Smart, so we can check that as well. I kind of have them grouped together, all the pulled questions from Chapter 1 and Chapter 2. So starting with Chapter 1, we went into the characteristics of life. So I've got the answers covered up, but you know it, so do it with me. Living things are organized, made of cells. They acquire materials and energy. They maintain homeostasis. They respond to stimuli and reproduce. They grow and develop, and they adapt and die. Yay! All right. We then started we talked about some other things, but I'm focusing just on big ticket items here. We talked about the 4 big ideas. So the big idea 1 was the process of what? Evolution is a key word, right? Drives the diversity and the unity of life. Okay, so we are looking for the patterns, and as you've got a few chapters under your belt now, and you want to start looking for that. Big idea 2 is that we utilize energy and molecular building blocks to grow. Utilize energy. Right now, you know, oh wait, have I got those reactions in here? No, we don't know them. You'll learn them. Utilize, sorry, I'm fast forwarding. Utilize energy. We started on that by talking about enzymes and energy, right? We're moving towards cellular respiration and photosynthesis, and how you can use that energy to grow, reproduce, and maintain homeostasis. Living systems retrieve, transmit, and respond to information essential to life process. What's some key information that our cells have? DNA. And biological systems interact, and these interactions possess complex properties. So we started talking about some of our important organic molecules, and here's a list of those, right? And we went into those 4 important organic molecules. I introduced them in Chapter 1, but we went into great detail with them in Chapter 3, right? Yeah, Chapter 3. So you can see at the top, you can see amino acids, and you know the basic structure of amino acids and how those build proteins. You can see a nucleotide there building a chromosome. You can see a carbohydrate right here, and it's implying that this carbohydrate is getting made in a what?
Plant cell.
Yeah, what part of the plant cell is it showing you right there? Yeah, chloroplast. So you would find in a plant cell. And then here you're looking at a fatty acid. Does this particular fatty acid look saturated or unsaturated? Saturated. So would it be a solid or a liquid at room temperature? Solid. Yeah, so it's showing you these are the 4 important organic molecules, and we already know those can be used to build organelles in cells, right? So now that will make more sense to you as we look back. And then these reactions right here, these are coming right here. And we're going to talk about cellular respiration, so that will look familiar too in just a little bit. Remember we talked about that all life comes from what? LUCA, which stands for the last universal common ancestor, right? And we're talking about life evolving from that because it has those same, uses the same molecules, same metabolic functions, same key molecules, ATP, DNA, RNA, all of those types of things. And we said, if they're living in an environment, what do they have to do if the environment changes? You're going to do one of 2 things. Adapt or die. That's it. Those are your 2 choices. So you're either going to adapt and be competitive or you will die. The mechanism would be natural selection. That would be selecting for an adaptation that is advantageous in that environment. Now, other organisms might live just due to chance, and that's called genetic drift, and we're going to be learning about that. But natural selection is the biggest mechanism for evolution, the mechanism to explain change over time. So that's all falling underneath big idea 1. Big idea 2 is cellular processes, energy, and communication. We're really starting to delve into those now. But the first part we talked about is probably how energy flows and nutrients cycle, if we look at this. And ultimately, all the energy, and we've learned this already, comes from the sun, right? And that energy gets passed through producers and consumers, and decomposers work on all of those to recycle the nutrients. And I believe, yes, I know we did. We introduced the laws of thermodynamics. The first one is that energy cannot be created or destroyed. It can only, yes, and every time it transfers, you lose some as heat. And we talked about free energy, right? And that's energy available to do work. Because we're not losing energy. It's not like, oh, I've lost the energy, I can't find it. I just lost usable energy. And what do we call that change in energy? What do you call it? Delta G. Yeah. And then we said most reactions, most metabolic pathways are controlled by what? You did a lab on it today. Enzymes, right? Enzymes catalyze reactions. You can see the significance of that, right, with our reaction that we did today. We could see the difference in just the substrate, right? And how the enzyme, how the reaction was affected. Another thing we could have done today is we could have set up hydrogen peroxide, and just instead of adding catalase, we could have added what to it? What would be a good, just adding plain old what? Water, right? To see that hydrogen peroxide will not break down unless it's in the presence of catalase, that enzyme, yeah. And what does it break down into? What are you measuring? Oxygen. Good. And what else does it break down into that we did not measure? Water. Good. All right. So we take in nutrients in order to maintain homeostasis, and we're going to learn the process of breaking down those nutrients through cellular respiration that are provided to us through photosynthesis or another organism that has consumed things that have done photosynthesis. Then big idea 3 is genetics, information, and transfer. And we have talked about DNA as being our code book. And we have already kind of, even though we haven't gone into those chapters yet, how does the information that's located in the nucleus, the DNA, get out? mRNA. What does M stand for? Messenger. And how is messenger RNA different than DNA?
It gives it to the ribbon.
It's single-stranded. It has, yeah, what is that? Has ribose instead of deoxyribose, and uracil instead of thymine, right? And RNA will make a copy of the DNA, and it'll go out on the workbench. What's the workbench? Ribosomes, good. And there you will, and we'll learn about codons and anticodons. That's all coming. Okay, you'll learn how tRNA, what does T stand for? What does it transfer? Amino acids, right? And we hook amino acids together to build a protein. So we can see the mechanism, basics of that mechanism on how DNA codes for all the proteins in our cell, right? And remember we talked about the protein and protein structure. So we have other proteins that help fold them and get them in the right shape. What are those called? You might need them on a date. Chaperone proteins will fold them. Yeah, right. Chaperone protein. Sorry. Chaperone proteins will fold them. Now, when we talk about protein structure, it just makes me think for a minute at the 4 levels of protein structure. Are you thinking about those 4 levels?
Yes.
What is the first level? Primary. And that is the sequence of amino acids in a chain. Who determines the primary structure of a protein?
DNA.
The DNA. Who did it get the message from? mRNA. Where did it get built? Ribosome. Who brought the amino acids there in that right RNA? Thank you. Okay. That's a primary sequence. What is this, our primary sequence, which is the primary structure? What is the secondary structure? What are my choices? Alpha helix or good. And what determines, like, what's making it go into those shapes? Charges on either end of the amino acids, right? You have an amino group and a carboxyl group. What's an amino carboxyl? What's like up top? H, and then on below is a R. Just in my analogy, yes, you could have them either way, right? How many different R groups are there?
20.
And those R groups are going to determine what structure? Tertiary structure, the folding pattern imposed upon the secondary due to the R group interactions, because some are hydrophilic hydrophilic, and some are hydrophobic. Right. And then the quaternary structure, not all proteins have that. That's when you have what? More than one chain, right? So we know when we look at the basics of genetics, it's the DNA that's determining the sequence of the mRNA, which is determining the sequence of the amino acid, which is giving us our proteins, which is what builds, the enzymes are proteins, right? So all of that is determined. Everything I see that's not your clothes, those are proteins that I'm looking at right now. That's what makes you. So the DNA codes for that, okay? That DNA even codes for our behavior, and we'll be learning about the connection. Some behavior is learned, right? Some behavior is innate, and we'll talk about that and how we respond to that in our environment. Okay, big idea 4 are the interactions. So if we look going from small to big, we said living things are organized, made of cells. So let's look at that organization. Remember we learned atom, molecule, organelle. Do all cells have organelles?
No.
Who has organelles?
Eukaryotic.
Eukaryotic. Who doesn't? Where could eukaryotic cells have come from? Living together, right? What do we call that?
Endosymbiotic.
Endosymbiotic hypothesis or theory. That's good. Endosymbiotic. So one prokaryote went to live in another prokaryote and be its organelle. Now that would explain things like mitochondria and chloroplasts, and there's evidence for that too. And the evidence would be that mitochondria and chloroplasts have their own DNA, and it is in the shape of a, which is just like prokaryotic DNA, and they have their own ribosomes. Yeah, it's like workbenching it, and their double membrane, right? So that explains those organelles. When we look at how things cross a membrane, how things come in and out, one of the things it can do is this. What is this? And if one of these ribboned around in there, maybe it could become the R. Yeah, endoplasmic reticulum, right? You have a big endocytosis, right? Now I was 1, now I'm 2 layers, right? You ribbon through, that can be the sorry, say it. Yeah, that's what I want, endoplasmic reticulum. And if that endoplasmic reticulum, ironically, is continuous with the nuclear membrane, so that ribbon could have just ribboned all the way around, what would that be called where the DNA is in a prokaryotic cell? Nucleolus is inside a nucleoid. Nucleoid, and ribboned around that to enclose it. That makes sense to you? Okay. And that could have been the beginning of the nuclear envelope. And our evidence for that is it's double. Yeah, all right, good. So atom, molecule, organelle, not all cells have organelles, right? Cell, then what?
Tissue.
Tissue, let's do it together. Organ, organ system, organism, population, groups of the same species, multiple species, the environment, abiotic factors, and where all living things exist. Good. Okay, as we're building up. All right, and so you have interactions between them. What do you call the traits that an organ has that are more complex than what a tissue has? What do you call those properties? And being a dolphin. Emergent properties. Yeah, you increase in complexity as you go up. Emergent properties. Do you remember that? Yeah. Okay. Then we started talking about the science practices that we do. Student can use representations and models to communicate scientific phenomena and solve scientific problems. Have you used any models yet?
Yes.
Models of a cell membrane, perhaps? What did you use to model cell membrane? Dialysis tubing. Yeah, I used a physical model to model a cell membrane. A student can use mathematics appropriately.
Chi-square.
Chi-square, exactly. A student can engage in scientific questioning, the driving, to extend thinking and to guide investigations with the context of the AP course. The student can plan and implement data collection strategies.
Data table.
Yeah. In relation to a particular scientific questions. A student can perform data analysis and evaluation of evidence. Yes. The student can work with scientific explanations and theories. Well, you've dealt with theories. There's the cell theory, right? The theory of evolution. Those are all theories. But that you can also do when you look at the student can perform data analysis. That's claim, evidence, reason. Good. And science practice 7, the student is able to connect and relate knowledge across various scales, concepts, and representations in and across all domains. Sure. Okay. He said peptide bonds. All right. Now, scientific method, you know, because every time we do a lab, right, you have a driving question, you have some background information that you need before you can write your hypothesis. We've looked at 2 types of hypotheses. What are the 2 types?
Research and null.
Research and null. So research says there's a connection between the independent variable and the dependent variable. And a null says there's no. But they may not always actually think there's no, but they want to see that it has an impact on it. So that's why you write a null, and then you see if the data is significant. Okay, exactly. Then once you have your data, you always have a controlled experiment, right? In all of your investigations that you've had. Then you can collect your data, analyze your data, and claim evidence, reason, and then come up with your conclusions as a result of that. And does it support what you said could you answer your driving question or not? So we talked about chi-square already, so that's good. All right, now I have a bunch of questions in here, but do you want me to go on and do chapter 2 right now real quick and then do the questions?
Yeah.
Okay. So chapter 2, that was the kind of easy peasy chapter because all it was is a review of the chemistry. And so we talked about atoms. Let's do the atom story together. The atom story. Atoms are made of protons, neutrons, and electrons. The protons and the neutrons are in the nucleus, atomic mass. The electrons are in energy levels. 2, 8, 8, 32. 8, 8, 8 is great octet rule. Why do atoms interact? They're trying to get happy. How do you get stable?
8 electrons.
8 valence electrons or fill your outermost level to the mass. Okay? And in order to do that, sometimes they're born that way. That's how they are. They're stable. What do we call that whole group?
Noble gases.
Gases. If not, you're going to enter into what?
Covalent or ionic bond.
Yeah, basically. There are other ones too, but ionic or covalent bonds. Which one is about ditching or stealing electrons?
Ion.
Yeah, so first you have to form an ion, and then you're attracted or repelled from each other, yes?
Yes.
Covalent bond is about sharing.
Sharing.
And when you share in pairs. Do you always share fair?
No.
What does sharing fair look like? What's that called?
Non-polar.
Yeah, non-polar covalent. And when you share unfair, it is polar. What is a classic polar covalent bond that affects all of life all the time? Water. Yeah, water. So water is a polar molecule because oxygen pulls on the electrons more tightly than hydrogen, which makes it partially positive and partially negative, which allows it to interact with those things that are hydrophilic. Yes?
Would diatomic molecules be non-polar molecules? Would they share?
They would share fair.
Okay.
Yeah. Okay, and we looked at how to identify how many valence electrons you have. We looked at how to put them in a periodic table. Remember, periods are on the horizontal. They're in the same energy level, right? And then groups have the same number of what? Valence electrons. And this is a truncated, right, periodic table. It's not a complete. You're going to learn more about that. But then that helped us to understand why you would form an ionic or covalent bond. We're good with that part?
Yeah.
Okay. And then we talked about water. And then we talked about the properties of water, right? And we talked about its high heat capacity, meaning it can hold a lot of heat because it shares it in all of those what? Hydrogen bonds. It has a high heat of vaporization because you need a lot of heat because it can hold a lot of heat. It takes a lot of heat before it actually evaporates, right? We talked about going the other direction. If we're losing energy, it becomes not hot but
Cold.
and it changes when it's cold. What does it do? It becomes more spaced out. Its hydrogen bonds less dense, so it will float, which is a biological benefit because it helps what life? Yes, it helps insulate life in bodies of water because The ice will be on top and the water underneath is below. Why is it a biological benefit that it has a high heat capacity?
It's, water is resistant,
Yeah, our bodies are stable. It also helps to what?
So we don't evaporate.
Cool us. It helps to mediate the effects of temperature in our environment when you're near a body of water because it can hold that heat and re-radiate it back out. Those are all true. Okay, we talked about this. We talked about that it's a good solvent for things that are hydrophilic. We talked about the properties of adhesion and cohesion. Cohesion is one water molecule with another water molecule being held together, right? And then adhesion is when those water molecules are what? Yeah, another polar surface. And the biological benefit we saw there was especially in trees to maintain columns of water where you're pulling the water up from the roots all the way to the tippy top, right? And what do you call that evaporation of water out the tops of trees? Or leaves? Transpiration. Good, transpiration. All right, we talked hydrophilic, hydrophobic, adhesion, cohesion. Then the last thing we do is talk about pH. And pH is a measurement of acidity, a negative logarithmic scale of the number of hydrogen ions, right? And so what we know in our body is we need to make sure that we have buffers, right? In order to keep our pH just right, because that can affect the structure of the molecules that they're in, right? Another way we could have changed this lab today is you could have done the same lab, 5 mils of hydrogen peroxide every single time, a half mil catalase every single time, and then we could have added increasing amounts of hydrochloric acid. What would the hydrochloric acid do? Stop it. How? Why would the hydrochloric acid change it? Because it would change the
pH.
Yes, which would affect the structure of the active site of the enzyme. It would cause an allosteric change in that enzyme. Okay? Good. And then that was the end of that. And I just put in a bunch of questions. Does anybody want to do some questions?
Yes.
Yeah.
Okay. So I don't remember, I just dragged every question one out. I don't remember what they are. So hello, Smart. And you know what? I'll stop these. I'll pause while they're doing it, okay? And so you'll see me just in a nanosecond here. Okay, here we go. I don't even remember what the question was. How'd you do? Yay! Okay, really hard question. Here's another one. One question, so I'm giving you a nanosecond. Is it a hard question? No. 5, 4, 3, 2, 1. Yes. All right, metabolism. Okay, here's your next one. It's just whatever I have, sweetheart. I know you're just appreciative for anything I can do for you. Okay. I'm going to pause just for a second. Okay, let's see.
Let's go.
Yay. Oh, why was A wrong? Somebody picked that. Molecule before cell and tissue before organ.
Dude, we're the only one on.
Okay, stop. Okay, this is one that nobody should miss, right? So if you're doing it to be, stop. If you're doing it to be funny, it's just a distractor because it makes me think that I need to reteach that. I shouldn't have to, right?
Yeah.
Okay, all right. Next, here you go. Just in case you're wondering, I'm going to move the quiz that's scheduled for Friday, then I'm moving it to Tuesday. Yeah, so you have the weekend to go over that. Yeah, I'll tweet about that. All right, come back to me. Ultimate source of energy is the sun. What is the mechanism called? Natural selection. Good job. What do you call a concept that joins together well-supported and related hypotheses? A theory. Okay. Quantitative data is measurable. Good job. Thanks. All right. Moving Here. Hello, smart. This is chapter 2 questions. Whoa. I'm going to pause it real quick. Okay, here we go. In. Yay! Yay! Oh. How would you know how many, what did they say here when they meant 18? What were they really talking about? Neutrons. That is terrifying. Terrifying. Okay, here you go. Ready? One question, 5 seconds, go. 5, 4, 3, 2. All right, here we go. Oh, ionic bonding involves all of those. All right, here's another one.
It's not covalent.
This is what? Yay!
Covalent.
One question, 5 seconds, let's go. Here we go. Okay, let's talk about A. You guys, one person missed it, so I want to talk about this part. Atoms of the same element that have the same number of protons, but different number of neutrons are called what?
No, I knew it.
You didn't do that? Okay, isotopes. Okay. All right, as long as you get it. Okay, here's your last set or sets. I don't know how many questions are in here. We'll see. Last one. You can always go back. Did I give you quizzes over these chapters? Oh, I can make some. 3 and 4. I'll make some on chapter 1. Last questions. Oh, here, let's get our. You ready? Here you go. Hi, guys. Sodium and chloride are important for other things in the body, but it's hydrogen for acid base, right?
Yeah.
And number 2, A. Yeah. Questions? Yes.
I guessed right, but why is it A?
Okay, so which of the best describes the changes that occur when a solution goes from a pH of 5 to a pH of 8? So, what would I don't even know where I came up. What would 5 be considered?
Acidic.
Acidic. And then this would be a base. So what happened to the number of hydrogen ions?
It went down.
Hydrogen ions went down. Who went up? OH went up. Hydroxide. Got it? All right. Have a piece of toast. Don't stay up too late. You're welcome, sweetheart. Love you.